everyone, welcome to Ratio Christi. It's great to see everyone here. My name is John Feltz, Associate Chapter Director, and right here we have Dr. Brian Miller. Our third guest speaker, or second, something like that. So I'm going to briefly introduce Dr. Miller. So Dr. Brian Miller is Research Coordinator for the Center for Science and Culture at Discovery Institute. He holds a BS in Physics with a minor in Engineering from MIT and a PhD in Physics from Duke University. He speaks internationally on the topics of intelligent design and the impact of worldviews on society, and helps manage the ID 3.0 research program, having been a primary organizer of the Conference on Engineering in the Life Sciences, CELS. He also has consulted on organizational development and strategic planning, and he is a technical consultant for The Startup, a virtual incubator dedicated to bringing innovation to the marketplace. He has contributed on multiple books and journals covering the debate over intelligent design, including The Mystery of Life's Origin, The Continuing Controversy, The Comprehensive Guide to Science and Faith, and The Inference Review. He is a regular contributor to Evolution News and Science Today and the ID the Future podcast. Everyone give a warm welcome to Dr. Brian Miller.
Well, I'll never actually get used to hearing your biography. Does that feel kind of weird when people do that? It's just, and then I always like people to kind of mix stuff up just to make it more interesting. Like I went to the moon, there was Mystery Universe, but that's fine. But it's really a pleasure to be here. And I'm going to be talking to you about origin of life, thermodynamics, and design. And I actually presented this material in a sort of a secret meeting with Nobel laureate physicists, top level organic chemists in the world, top mathematicians. So you're going to get like, sort of cutting edge research. And literally, I've been doing reviews of articles that I can mention to you in passing too. Now, it sounds like there's a lot of physicists. Who are the physicists in the room?
Oh, yeah.
We got some physicists. Who are the engineers?
I'm a minor.
Okay, so we got the linguist. I'm really excited about that. And what were your majors, the remaining people?
I'm a marine biology major.
Oh, cool. Very cool.
I studied theology.
Oh, you do? Oh, are you a grad student in theology? Or?
No, I did that in the past.
Oh, okay.
What about
Oh, nice to meet you. It wasn't just a bunch of students running loose. It was like, I'm glad you have a character. Now keep them in line. So, where did you study theology, by the way?
King's Evangelical Divinity School in England.
Oh, gosh. Crossing the Europeans, that's awesome. Okay, and what I'm going to do is I'm going to break this talk into 3 parts, and after each main part, we can have conversation, dialogue, and question answer. It's a lot of material. But did you ever see like the Karate Kid, the old version with Mr. Miyagi? Remember at the end you have like the crane kick. It was like Mr. Miyagi said there is no defense. In the second movie there was, but that's beside the point. If you're dealing with issues of evidence of design, this argument is really as strong as you're going to get. I mean, it's about as rigorous scientifically in terms of both the positive evidence for design and also the negative evidence against a natural process. So if you can get this down, there is no defense, except for a multiverse. That's literally what I got. A Nobel laureate physicist said, well, maybe if we're in an infinite universe, the probabilities aren't going to be a problem.
That's what we talked about last time.
Oh, we did?
Yeah.
Who did that talk?
Me and John.
Oh, good for you. Yeah, well, then there you go. I don't have to deal with it. Thank you.
Rousing post-topic discussion on Boltzmann brains.
It was.
Oh, yeah.
Yeah.
No, but that's really the problem you get into. You lose rationality in science. So that's what you have to do to get rid of the design of the evidence of design. Now, as you who are interested in philosophy or theology know, that this design debate is not recent. A lot of people think it started with Darwin or the Scopes monkey trial, but this goes back to like half a millennium BC between people like Democritus and Aristotle and Plato, where you have like Democritus, who was a materialist. And by materialist, I don't mean someone that likes bling and money. What I mean is someone who believes that all there is matter, energy, time, and chance. So the materialists believe that there are these eternal atoms that would interconnect in different ways. And the rules of the interactions, chance, time, would explain everything we see from rocks the philosophers have thought about rocks. Versus people like Plato, who believe there is a mind, or Aristotle, who believe there is a mind, although Aristotle put the center of design within the organism, but I don't get into that. But the point is, there was a major debate in the 5th century, and that debate carried all the way down to the 1st century, where Paul has the famous passage in Romans talking about how the evidence of design is manifest in creation. And at that time period, in the 1st century AD, you have people like Epicurus with the Epicureans, and they weren't people that liked cooking. They were people that were following the tradition of Democritus. So they again believed They even had a primitive idea of natural selection from the poet Lucretius with an origins theory. And what's interesting is the Greeks even had an idea of adaptation and heredity that was virtually identical to Darwin's idea of pangenesis. So these ideas are not new. While the Stoics had ideas of design that carried over from like Plato and Aristotle. So this debate is nothing new. So when people talked about people suppressing the truth, he was engaging the scientific materialists of his day. So again, nothing new under the sun, as you see in Ecclesiastes. Now, when you talk about the origin of life, what you have is many different ideas of how it came about, but there's sort of common themes. And to get life, you have to have some sort of primordial soup or thermal vent or some environment on the Earth where you have some source of energy like lightning or proton gradients or heat, and you produce the building blocks of life. And the most important blocks are like your simple sugars, your fatty acids, your amino acids, and your nucleotides. These are actually really hard to build, and you need these. Eventually, these building blocks formed into larger macromolecules like complex sugars, cell membranes, proteins, DNA, RNA. Eventually, they were encapsulated in some sort of cell membrane. Eventually, metabolism originated. Eventually, you get everything from protein translation and eventually cell division and full self-replication with an autonomous cell. Now, the order will differ depending upon the theory, but those are the basic steps that you find. Now, the first step is where things fail, actually. I mean, because I'm sure you're all familiar with the Miller-Urey experiment, where you have like that famous experiment where you take gases which were believed to have been on the early Earth and you shock it, and then what happens is you produce some amino acids. And the first version of the experiment, actually they realized the gases weren't right, so there were later versions that were less reducing. But what you'd find in these experiments is pretty much always the same. It doesn't matter what you do. You take any organic material, you put any energy source, any conditions, you get tar, you get gunk. You get maybe a few amino acids, if you're lucky, some glycine because that's the simplest one, maybe a few others, and that's it. That's it. And the problem is these building blocks are in a very complex mixture with many, many other molecules. So what you'll never see is going from this stage to this stage because there's so many other molecules that this will interact with. End of story. So what you find is all of the origin of life experiments that use realistic conditions end at that point. So what you have is people kind of gave up on those realistic conditions, and you have more synthetic experiments. What happens with these experiments is you have incredibly complex protocols where you have certain molecules, you have certain reagents, certain conditions, you produce a little bit of what you want. You then will multiply it, concentrate it, have different conditions, different reactions. You get a little bit of something you want, and you do this like multiple times. And through all these different stages, you eventually get like a precursor to a nucleotide. So in all these experiments, RNA world, autocatalytic cycles, it doesn't matter, you only can advance with enormous levels of investigator intervention. That's key. And you take any of those experiments, you go back in time, you dump it into a thermal vent, you dump it into some pond in the middle of a cave, doesn't matter. What happens? The system collapses and it breaks back down to simple chemicals. End of story. Now another issue, and in fact, this is such a great quote. It's by Robert Shapiro. He was one of the leading origin of life experiments decades ago. He's passed away, but he said this about these experiments that produce like nucleotides. The analogy that comes to mind is that of a golfer who, having played a golf ball through an 18-hole course, then assumed that the ball could also play itself around the course in his absence. He had demonstrated the possibility of the event. It was only necessary to presume that some combination of natural forces, earthquakes, winds, tornadoes, and floods, for example, could produce the same result given enough time. So you see the analogy, right? So it's a very different situation when you guide everything step by step and versus allowing everything to happen purely by chance. Now another issue, which I really have never seen, it's rarely talked about, is the localization problem. Because imagine you've got 1 liter of water, using metric for people from the UK environment. You have one cubic meter of water. What happens? Imagine you have a cell membrane and you have a protein. And now what has to happen is they have to find each other, right? If you're just dealing with diffusion, it could take millions of years before they make contact. So the problem is this protein is going to break down long before it even makes contact. Now, when you do with origin of life, the big problem is that the different building blocks require different conditions, different temperatures, different pHs, different environments. In fact, there's a great paper I have that has, I think, at least 9 different environments are needed for the building blocks and linking them together. So now we're not talking about 1 liter of water, we're talking about random locations on the Earth that somehow have to localize and find their way into a cell membrane. So this problem of localization is insurmountable. There is no way around this localization problem. But that's just kind of a warm-up, because I'm going to talk about the real problem. And this deals with more of the thermodynamic challenges to the origin of life. And when you read critiques of origin of life, let's say the 1980s, a common critique is that life is simply too improbable to come about. That there's just so many more ways you can arrange molecules to not have life than to have life. And one of the famous people that talked about that was Fred Hoyle, who came up with the famous analogy that even if you have all of the building blocks of life in a test tube or a beaker, the chance of them forming the right interconnections is like 1 chance in 10 to the power of 40,000. He said that's kind of like the chance of a tornado going through a junkyard, throwing a bunch of debris up in the air and having a 747 jet airplane fall to the ground filled with gas ready to fly. Can't happen. You've all heard that analogy? That's just great. That's nostalgic. You have to remember that analogy. And as a side note, what you have to recognize is you who in the physics know is probability is very closely associated with entropy. Because if you're looking at, let's say, a system, and you have this, what's called a macro state, where you've got 2 balls on one side and 2 on the other, versus other microstates that form different macrostates, like here you got 3 in one, and here you got 4 in one and 4 in the other. If you were to randomly select one of these possibilities, most of the time, or the majority, or the plurality of the time, which of these macrostates would you get? How many balls on each side?
2.
2 and 2, right? So what happens is this has higher entropy, because if a state has higher entropy, all it means is there's more ways for that state to come about. There's more arrangements of molecules that associate with that state. So entropy is very closely associated with probability. So when people say that life is really improbable, it's the same thing as saying is life shouldn't really come about because you're going from a state of high entropy to a state of low entropy. You're going from a state that's highly probable, lots of random molecules, to a state that's highly improbable, which is life. And people say that that's not possible, but that is not a complete argument, because there's one problem. There's a loophole that people don't think about. And that's that you do have low entropy states that form. Because if you take water and you put water at like a really cold temperature, it freezes. Now what has less entropy, water or ice? Ice, but it forms spontaneously, right? So there is a way around the entropy problem. And the reason ice will spontaneously form is because the hydrogen attracts the oxygen and it pulls these atoms together. So ice represents a lower energy state, or lower enthalpy state if you're a chemist, than liquid water. So that's a second driving tendency. Yes, it's really the case most of the time that nature wants to go from low entropy to high entropy. That's typical. But it also wants to go from high energy to low energy. Water runs downhill, it doesn't run uphill. It's kind of like if you were to roll dice. A less probable combination of dice would be like a low entropy state. Like let's say I have 10 sixes. That's very unlikely, right? Now imagine I came to you, you didn't know me. You didn't have John, backing me up. And I said, I will bet you $10,000. I can roll, I have this dice here, these 2 dice. I can roll 100 sixes in a row. I'll bet you $10,000 I could do it. Would you take me up on that bet?
I don't think I'd trust your dice.
Yeah.
Bingo. And that's the problem. The dice can be loaded. So the dice are loaded in nature for low energy states. That's key. So you can get low entropy with loaded dice. But there's a problem. Life is both low entropy and high energy. So what happens, it's kind of like trying to roll 100 sixes in a row if the dice are loaded for rolling ones. It's really a problem. And for those of you in chemistry or physics, you know that often you can combine the enthalpy or the energy with the entropy and get what's called free energy. And free energy is essentially the amount of energy you have that can do work. So there's lots of energy in the air, but that energy isn't going to drive you home, right? But you've got gas, and that gas has free energy because that energy can be used to do work. So all natural systems will go, in normal conditions, from high free energy to low free energy. But life requires chemicals to go from low free energy to high free energy. And there was a famous physicist named Harold Morowitz. He said the probability of some chemical soup forming a cell, like an E. coli, is like 1 chance in 10 to the power of 100 million. It's never going to happen. That's a real problem. But again, there was a loophole. Because typically if you talk about free energy, you're dealing with systems near equilibrium. You've got a constant temperature, you've got pressures you can measure. But what if you're far from equilibrium? What if you shove energy into your system? And a classic example of that is a tornado. So you have temperature gradients, you've got pressure gradients, and this non-equilibrium system self-organizes. Another example is if you boil water, you've got heat below, it cools from the top, you get roll patterns. You've seen the roll patterns in your spaghetti, right? That's called self-organization. If you repeat people like Ilya Prigogine, he was a physicist who argued that maybe the same way you get this self-organization in a tornado, you could self-organize life if you're far from equilibrium. Because when you're far from equilibrium, the physics is much less understood, it's much more complex, and maybe rules apply. That was the idea. Well, there's a major problem with that. Is that if you look at these that was kind of my physicist, my PhD, is I worked with these non-equilibrium dissipative systems. This is how I got into this, actually. I realized that people are going into my territory in ways that were not necessarily accurate. The problem is, if you look at a tornado or boiling spaghetti, the order you see is completely different than the order in life. Because the order in these non-equilibrium systems, like a tornado, are dynamic patterns, or patterns that are in motion. It's macroscopic order, it's more like on length scales that you can see. It's a spontaneous process. You're going to lower free energy, and it's a law of governed patterns. But the order in life is completely different. The order in life is more of a structural order. You've got molecules in fixed orientations. You're actually decreasing entropy at the microscopic level, while in all these non-equilibrium systems, you're increasing entropy at the microscopic level. You're generating entropy. And you have a higher free energy state, and you have specified complexity, meaning you've got information in the system. So the problem is the order in these non-equilibrium systems is completely the opposite of the order you need in life. But there's more sophisticated equations that came out, I think in the 90s, called fluctuation theorems. And Jeremy England is a physicist that really popularized this in terms of origin of life. Who's heard of Jeremy England? Anyone? Okay, that's fine. If you were into origin of life, you'd know Jeremy England, because he looked at these fluctuation theorems. And what he did was he actually talked about the probability of a system going from one macro state, like prebiotic soup, to another macro state, like life. And he found that the probability drops exponentially with the total change of entropy. And what happens if you look at the probability of a prebiotic soup becoming a cell, it's still like 1 in 10 to the power of 100 billion. So it's no more likely away from equilibrium as it is near equilibrium. So this is a major challenge. And let me just use an analogy for those of you that are not in physics. So I'm going to speak to all you who are not in physics. Let's use an analogy. People say all you have to do is you throw energy into a system and you can get really good order, right? Like a cell. But it's like if you have a library, and let's say half the books are on the shelf and half are on the floor, and low entropy would be like the books in the right alphabetic order or Dewey Decimal System or whatever system they're using. That's like low entropy, they're in the right order. And low energy is like, or high energy is like the upper shelf. So a cell is like all the books on the upper shelf in the right order. And you say to yourself, well, I just have to add energy and I can create this sort of order. But is that going to work? If there's an earthquake, will all the books jump to the top shelf? What's more likely to happen? The books on the top shelf are going to drop to the bottom shelf. It is possible, sure, it's possible, that a book on the bottom shelf will jump to the top shelf. It's possible, it can happen, you have energy, but that's not what's going to happen most of the time. And will this create more order or more disorder? And that's the problem. So the origin of life, arguing that simple chemicals are going to form a cell, is like arguing for a perpetual motion machine. It's a physical impossibility. Okay, that's part one. We're going to put it on pause. Any questions, comments, things you want me to explain further? Are you doing okay? Is this making sense?
Are there responses to this that people have?
Yes. That's
Is
going
that
to
part
get.
two?
Yeah, that's part two.
Oh, I know.
Yes. Yes.
Is one of the responses, why isn't the energy just a more organized energy? 'Cause when you suggested energy, I didn't think of an earthquake. I thought of I could expend energy to put them back in the right place.
Okay, that's an excellent question. This is exactly where Jeremy Lynn is going with this. But let me ask you this. Okay, let's ask about this. Okay, so let's say it's not an earthquake. What other type of energy could it be? Natural energy. What would be other examples of energy that you could throw in your library?
Heat, fire.
Fire. Yeah, you could
Burn the library.
you could set the couch on fire. That would work. Any other ideas?
Someone-
Ashes would rise, right?
Someone going in and putting the books right in place.
Okay. This is what we're getting at. So you're absolutely right. Is people? This is where we get into this idea of matching of energy to systems. But the problem is, and this is sort of a trailer for the next part, is the energy sources that you have on the early Earth are not matched to the order you need in life. In the same way, there is no natural process that's going to put those books in the right order on the top shelf. There's lots of energy sources you could imagine, but none of them are going to do what you need to do because they're not matched. You have to direct energy in the right way to produce the right order.
What do you mean by matched?
So for instance, if you have a car, you pour gasoline on your car and you set it on fire, is that going to help you drive across the country? Now, the engine matches the gasoline to the wheels. How does it do that? It converts the energy of the gas to mechanical energy, which is directed to the wheels to make them move. That's matching. So now you, what's that?
So is this like a localization problem then? Or?
Localization is definitely part of it, but it's more of going from a raw energy source to a secondary energy source. So your gasoline is raw energy. It's something from the earth. I mean, it is processed. We won't worry about that. Coal factory, whatever. But the challenge is you've got to have a machine, an engine, that converts raw energy into a secondary energy form that's accessible to your system. So when the engine turns it into mechanical energy, now that's accessible to the wheels. That's what I mean by matching. Okay, any more questions? Can we go on? All right. If this is getting too boring, you want to walk out, that's fine. I'm not easily offended. It's sort of a job criteria to not be offended by people yelling at you and making fun of you. It's just one of those things. So there's only one way to overcome this problem, and that's machinery and information. This is the only way. You remember? You ever see like the End Game with Thanos and remember what did Dr. Strange say? One way. So there's only one way to solve this problem. In the same way, there's only one way to defeat Thanos. So here's that one way: machinery and information. And to use the analogy of the house, if you hear all you need is energy to order your house and you set the couch on fire, is that going to help? Not so much. So you think to yourself, well, that's a problem. So I'm going to get a robot. I'm going to buy a robot off eBay or off of Amazon. I'm going to plug that robot in. And what the robot does is it converts one form of energy, like electricity, to a form that's useful to order your house, like mechanical energy. So you buy this robot off eBay, you plug it in, you power it up, and then you turn it on, you let it go. Is that going to help? What's your robot going to do, most likely?
Bump into walls.
Yeah, it's not going to help. You're going to break things. Because what are you missing? What do you need?
Purpose.
Tell them what it's supposed to be.
Said, yeah.
Yeah, what do you call that?
Instructions.
Instructions, a program. So it's the information. So that's what happens without information. But if you have information, you say, robot, walk 3 steps, take your vacuum, do this. Go to dishes, do this. So you have to have instructions on how to order the house properly. You know, thinking like wax on.
Yeah.
I see where you're going with that. So you have to have instructions. It's the exact same thing in life. Life has very complex machinery. And what this machinery does is it takes the energy in the environment. It could be sunlight, it could be glucose, it could be some phosphorus chemical, doesn't matter. and it converts it into a form of energy that's useful for life. Now, the biology person, what are some of those useful forms of energy that the cell creates?
ATP.
That's a classic, ATP. That's probably the most popular. You get NADH, different things like that. So they're called energy currency molecules because they're molecules that act as batteries for your cells. It's what gives power to it. So you have to have this machinery that converts the energy available to the energy that's useful for life. Without that, you're dead in the water. Raw energy of any sort from the chemicals or the energy sources will destroy a cell in the same way a fire is going to destroy your house. So you have everything's tending towards equilibrium. What happens, these engines, like ATP synthase, produce these high energy currency molecules that power your cell and create homeostasis. Homeostasis is just a state that's stable but far from equilibrium. That's life, it's homeostasis. So what people have done is they said, what possibly on the early Earth, this is the response, could give you these free energy solutions, these high energy molecules. And the most popular model is thermal vents. So what happens, you have some alkaline thermal vent on the ocean. You've got an acidic ocean. You've got a basic interior. There's kind of this sheath or membrane that forms over some crevice in this alkaline vent. The proton flow can convert one molecule into a higher free energy molecule. This has been done. Nick Lane is one of the leaders of this, as well as Michael Russell. And what they did is an experiment where they were able to create what they call a reactor that's kind of like a thermal vent. So that what they do is they're able to produce these free energy molecules, higher free energy molecules. Now the question becomes, how much energy is necessary just to maintain a cell? So what happens, based on this paper and others, what you find is that if you're going to go to like a cell, like E. coli, what happens is a typical cell is producing like 1 watt per gram of power of high free energy molecules to run your cell. That's how much a cell produces. That's like a sports car. That's a lot of power. Well, just to maintain a cell, you're going to need about 50 million nanowatts per gram. Nanos 10 to the negative 9, or 1 billionth. The problem is these thermal vents produce about 1 nanowatt per gram of free energy. You see a little bit of a difference here. So this is just to maintain a cell. This is just to make sure the cell doesn't break apart, so metabolism doesn't crash and it comes going back towards equilibrium. So the most you get from these thermal vents or other sources is minuscule compared to what you need for a cell just to maintain itself. So as you go away from equilibrium, what happens is you have to produce more and more energy just to fight the thermodynamic forces pulling you back towards equilibrium, because molecules are breaking apart, they're unfolding, you're having to create more of a disequilibrium between the outside and the inside. So the problem is the energy you need is so much more than what you can provide from natural processes that you have a serious problem. But there's another problem. In these thermal vents, you can create formaldehyde. Ooh, so you take carbon dioxide, you increase the energy to formaldehyde. Well, you produce like maybe 100 billionth of a mole of formaldehyde. And if you look at experiments to produce things like the precursors of nucleotides, that's like more like half a mole. So the concentrations you produce are so small, it's useless, dead in the water. And there's other, Helen Hansma is another person who talked about mica sheets. I actually had a very nice conversation with Jeremy England in the journal Inference. He was very respectful, he's a really good guy. But then Helen Hansma said, well, maybe these mica sheets can help. Well, the problem is, the idea is you've got these mica sheets and they push molecules together is the idea. So the mechanical motion helps you to create high free energy molecules. But how often is that going to happen? I mean, if you have that happen like in a square centimeter, maybe once every second, that would be amazing. But you need more like billions. So again, you're not going to get anywhere near the energy you need. So this is a huge problem. And what happens is all these origin of life experiments where they produce maybe a nucleotide precursor, is what they do is they do things like they add reagents, they purify, they remove things. This is increasing the free energy. So all the origin of life experiments, through the investigator intervention, play the role of this machinery of life to increase the free energy. So it's very artificial. So that's a big problem. But now, that's just the energy. Now the problem, though, is still the issue of probability and the entropy, because this is a really nice paper by Tompo and Rose, and it's called the 11th law paradox of the interactome. If you take a yeast cell and you just look at the possible ways that proteins can interact, the number of ways is 10 to the power of like 80 billion ways these proteins can interact. So the probability that, and let's say the first cell is maybe a tenth the size of a yeast cell, or even a hundredth, you're still seeing the probability of random molecules forming a functional cell is like one chance in 10 to the power of a billion. It's never going to happen. So how do you get around this problem? And the answer is with enzymes. Because if you look at what enzymes are, they drive chemical reactions. So this is a typical metabolism, and this is a citric acid cycle in metabolism, a very primitive, very standard metabolic cycle. And the way these cycles work in life is every single chemical step in life is managed by an enzyme. And an enzyme is a long chain of amino acids. And the chain has to have the right sequence of letters in the same way a sentence has to have the right letters in the right order to make sense. So you have information in these sequences that follow in the right structure, and then they will drive the right chemical reactions. So in the same way, your computer program is what allows your robot to do what you need to do to not destroy your house, but to clean the dishes. Enzymes contain the information needed to drive the right chemical reactions to run a cell. So information is essential. And what is information? Information is anything that constrains possibilities or reduces uncertainty. So if you have Google Maps and I tell you that there is a buried treasure, and I'm going to give you the location of that treasure, and you just have to find it. It's just buried a little bit below Earth. And I tell you it's at 22 degrees north and 114 degrees east. Does that encourage you or does that discourage you?
It's pretty big.
Because there's not much information, and there's so much of an area. But what if I give you more decimal points? Because every time I add decimal points, that's more information, and I constrain the search area until it becomes very searchable. So information constrains possibilities. It reduces uncertainty, and that's really key. And that's what you need in life, because enzymes and proteins are chains of amino acids, and there's 20 amino acids in the same way there's 20 English letters in the alphabet. And these sequences of amino acids have to be in the right sequence, or else you're not going to fold into the right structure to perform some useful function. And the chances of a random set of sequences, a random sequence folding into a simple enzyme is like one chance in 10 to 100. That's like picking one marked atom in the entire visible universe. It's so rare it will not happen by chance. And again, the information in the sequence is what allows these enzymes to reduce uncertainty or to constrain outcomes, taking causal control. Now, what you see in origin of life experiments is all these complex protocols reduce uncertainty. They constrain possibilities. There might be lots of way reactions can take place, but all these complex protocols, all these steps, force outcomes. So the information in the protocols plays the same role as the information in those amino acid chains that form enzymes, because the information is what forces the outcomes that you want. So in origin of life, people use all these complex protocols to produce what they want. They're demonstrating the absolute necessity of information to move molecules towards life. That's a key point. So again, these protocols act the same way as these enzymes. Now, I mentioned Jeremy England. He had a really nice book called Every Life on Fire. And what Jeremy England did is he described in really beautiful terms the challenge. And the challenge he describes is that life is always breaking apart. So you have to have machinery that's pushing life back up the hill to higher free energy. So you always have to refold proteins. You have to maintain metabolism. So you always have to use energy in these machines to maintain life. He talks about this. And he does a beautiful job of talking about it. But he also talks about a big challenge is matching. This is what I talked about, is that you can have an energy source, but for it to produce some interesting result, the energy has to be matched to your system, just like we talked about, just like the engine matches the gasoline to the wheels by converting gasoline into chemical energy. And the problem, as I mentioned before, is when you look at all the energy sources on the early Earth. They're not matched. They have no connection to anything useful for life. So this isn't going to work naturally. You have to have enzymes that make this happen. And what happens is Jeremy England refers to these different experiments. Like you might have these colloidal spheres, and you might produce like an electric current through them or a magnetic field, and you get cool patterns. And the reason you got cool patterns is because the experiment is designed in such a way that your energy source is designed to match and interact with your system in a way that creates interesting patterns. It doesn't happen naturally. So every experiment that England referred to, others referred to, are experiments where the energy source was designed specifically to match with the system to produce the results they wanted, or something in the same category. And usually it's the same material. It's the same beads. It's the same spheres. So the matching is easy. In life, though, what happens is there are hundreds of chemical reactions that are completely different. They need different conditions. So what happens is these enzymes create the right microenvironments to drive the right conditions, and these enzymes will also take energy from these high energy molecules and direct them towards the reaction you want. But you need a separate enzyme for every reaction because they're completely different. So you have to have different enzymes to access the energy from the high energy molecules to drive these reactions in the right way. So again, it gets back to information. And this is a great paper by Yasser Demirel. And again, he says, information in biological systems in the fluctuation theorem. He talks about, this is a great quote. He says, he talks about how if you want to reduce entropy, you have to have mass, energy, and information. This is the key point he makes, that these are essential. And again, why is this important? Because information always points to a mind if you have a lot of it. Now imagine that you're sick, and you go up to your room, and you come back, and you find this on your desk. Drink plenty of fluids and rest until you are feeling better. Could you explain this from the chemistry of the pasta? Like, is it possible that we will one day learn how the pasta, the organic chemistry of pasta, can create this? Will that ever happen? What about the physics of the boiling water? Will the physics of the water ever explain to you how this came about? No, because the information, the arrangement of letters, transcends the physics and chemistry of the system. It's imposed upon it. There's no relationship to it. So whenever you see large amounts of information, it always points to a mind, without exception. And I'll just kind of, these are amazing quotes, I'll just skip it, just because I'll kind of go into the question and answer. So let me just stop right there. So the big point of the last section is to create life, to maintain life. You've got to have machinery that processes energy and information to produce the state that you want. Okay, now any questions or comments about that? Is this making sense? Are we okay? Either it's so trivial, like, oh, was he? Right? That's so obvious. Or, what is he talking about? I don't understand. So I'll hope for the best.
Yeah, no, at least for me, a lot of this is like the things that made sense and that I kind of had suspicion on, but I'm glad that there's actually like hard things to back it up.
Yeah.
Then that's mostly it.
Yeah,
And just enjoying it.
And by the way, you asked what's the response. In fact, let me just actually read this because it really is interesting. This is a very famous paper. Michael Russell, he's a real leader in the origin of life field. And here's what he says. He says, we claim in particular that it is untenable to hold that life-relevant biochemistry could have emerged in the chemical chaos produced by mass action chemistry and chemically non-specific energy inputs, and only later have evolved astonishingly specific mechanisms. So what he's saying is that there's no way chemicals on the early Earth with random energy sources could produce life. That's shocking. And this is just a secular origin of life research says that molecules will never move towards life. 65, 70 years of research has shown that exact same. Every origin of life experiment ever done shows that molecules do not move towards life without intervention. But what is his response? Okay, this is like a thermal vent, a bunch of chemicals. Here's his response. Here's the savior. Instead, we claim it had to have been launched simple and specific, that's the chemical soup, and thereafter have been forced by the scythe of natural selection to maintain the necessary specificity standard at each evolutionary increment of complexity. More specifically, all the devices of life, metabolic and structural, must have been invented by it, in situ, step by step, and while in flight, starting from the simplest possible inputs. And learning itself by trial and error, which small changes, themselves occurring by chance, were useful, each incremental step in this evolution by creeps, complexity building process, necessarily being vetted by the system, both for fitting in and contributing more utility to cost. So what they're talking about is you've got this soup in a thermal vent that's never going to move towards life, but natural selection will select, will choose, will vet, will kind of pick out the right things and move towards life. Now, anyone see a problem in that logic? Yes.
Survival of the fittest assumes that there's already something that's surviving.
Bingo. You've got to have self-replication, or else it doesn't even make sense to talk about natural selection. This is just a bunch of liquid. It's soup in a thermal vent. There is nothing reproducing. So what happens, and I see this all the time in origin of life research, is they invoke natural selection, not as a real process, but almost like a demigod. It's like the demigod natural selection steps in and picks out all the right molecules and kind of puts everything together in the right way to get a cell. I mean, it's nonsense, but this is what you see all the time. So no, there is no real response to this. Okay, so now, any more questions or comments before we get to the last part? Yes?
Couldn't you hypothetically have something that wasn't a cell but still replicated, something a lot simpler?
Great question. So the most popular theory is what's called the RNA world hypothesis. And the idea is that you've got these RNAs that appear on the early Earth. I literally just wrote 3 articles about this, critiquing different research. It just came out in the last week, a few weeks. So you've got these RNA molecules, and what they do is they start, that one of them kind of folds into what's called a replicase or a ribozyme. So you've got this enzyme-like thing from this folded RNA molecule, and this thing goes around and starts replicating lots of stuff. So now that you have all these molecules replicating each other, you get a natural selection. So the system is selected for greater complexity, and eventually you get life. That's the idea. Yes, that's how they go about it. But there's a problem with that. There's actually multiple problems with that. The first problem is you're never going to get a self-replicating molecule. Like if you look at the experiments to produce RNA, that would be like Sutherland's, a classic example, in UK. You look at his protocols to produce an RNA precursor. They're incredibly complex. They use like vacuum chambers and changing reagents and molecules that you'd never see on the early Earth. So the conditions in these experiments are not even remotely similar to what could have happened on the early Earth. And even if you had a few nucleotides, they can't form chains because there's so many other molecules that interfere. But let's ignore that. Let's assume you have an RNA chain. When you put an RNA chain in water, it breaks apart. End of story. So what you have to do is you've got to take RNA chains and you have to give it the right information. So what they'll do, like Joyce is a classic example of this, is they'll borrow RNA sequences from life, like what's called a replicase from life. So they borrow the sequence. Okay, great. Now you have the right sequence. It folds into like a ribozyme. Well, you're adding information, right? So that shows you have to add information for it to work. But there's a problem still. The problem is these replicases won't replicate themselves. What do they replicate? They replicate nice, straight chains of RNAs that don't do anything. So for an RNA to do something useful, it has to fold into a 3-dimensional structure, but when it does that, it can't be replicated because it's in a tight 3-dimensional structure. Let's ignore that problem. You still have the problem that even the ones that replicate are error-prone. So at best, if you cheat a lot, you might get 98% accuracy. Well, that means that every generation you lose information, the system crashes because you get into what's called Eigen's paradox, where the information loss is so great that you constantly lose information. Let's ignore that problem. Here's the real problem. To get this to work, even to do like simple replication, you've got to have not one, but billions of these near identical molecules in a volume that's smaller than a drop of liquid. Now, you're not going to get one RNA in the entire history of the Earth. To get billions of RNAs with roughly the same sequence that happen to fold into replicases is beyond ridiculous. But the problem is if the numbers drop below billions, then the rate at which RNA degrades is greater than the replication rate, so the system crashes. So but let's ignore all that. let's imagine you've got this beautiful self-replicating RNA system. It's being selected for replication. So it's going to move away from life because as it gets more efficient at replication, it becomes less efficient at doing anything else. So no, it's not going to help. So that's a problem. And interestingly enough, James Tour, who's one of the top organic chemists in the world, we have a really, he interviewed me, we had a really great conversation. He was paid to do this. He was paid to create a self-replicating molecular system. He couldn't do it in a laboratory with all his synthetic organic prowess because the problem is it's really hard to get accurate replication because it's so error prone. So these systems will, in life, you know how we do it? In life, you've got like a DNA polymerase, but you've got error correction mechanisms. So what happens is the polymerase will do it only so well, but then you've got things that follow behind it to correct the errors. So that's the problem. So yeah, that's a big issue. Any other questions?
With that, do they argue that like sometimes you'll still get an enzyme with the incorrect folding, and so like those combinations of errors will result in something positive?
Yeah, that's the idea. Is that if you have it's really yeah. So any computer programmers in here? Now, if you start making random errors on your computer, is it more likely you'll get an Excel spreadsheet or it'll crash?
It'll crash.
Yeah, this is the problem. This is the problem. But that's what they argue. So you have high error rates. So you have lots of variation, and you'll get more likely to be able to explore sequence space and get something useful. The problem, though, is even if you happen to get something useful, in a few generations you've lost it. And the probability of a random sequence forming a replicase is just like you need about trillions of sequences just to do something really simple, like to bind to something or to ligate something. That's really simple compared to like replication. So you're never going to stumble across it. But even if you did, high error rates, you lose all your information. So, but they do argue that. Any other comments or questions? Okay, so let's talk about the positive case for design. What happens, and this is I'm actually doing a research project on this. I wanted to publish this in the next few years. There's lots of research on what are minimally complex cells, like how simple can a cell be before it stops working? And there's many different approaches. Here's just a picture of the different approaches. You've got more like top-down, bottom-up, more experimental, more theoretical. Lots of different ways that they go about doing this. And what happens if you compare those to theories of self-replication for engineering? Like von Neumann was a famous mathematician that talked about the theory of self-replicating automata, and he came up with criteria of what are the minimal complexities required for self-replication. And people apply this to things like NASA asked the question, how hard would it be to have a self-replicating space probe? It's really hard. And also you've got people who have thought about self-replicating robots. And when you look at these theories of self-replication, like what are the minimal components necessary and what are their interrelationships, you compare it with studies of minimally complex cells, overlap, very clear overlap. So if you want to have a cell, the simplest possible cell that can self-replicate, you've got to have energy production delivery, information processing, selective gateways with active transport, sensors, signal processing with algorithmic control, control systems, manufacturing, auto assembly, automated repair, waste disposal and recycling, global coordination. Essential. You miss one of these components, you're not going to be able to replicate. In fact, these are necessary just to exist. You don't have one of these things, your cell stops working and it breaks apart into simple chemicals. This is the problem. The information content associated with all this is well above 1 billion bits of information. It's like at the absolute minimum. It'd be like that amount of information in a Christmas Carol. So what you're seeing is the minimal amount of information necessary for a cell that wouldn't break apart into simple chemicals is enormous. So what you're seeing is evidence of design which is indisputable. In fact, did you see the movie Contact with Jodie Foster? Oh, you're too young. That's so embarrassing.
Parts of it.
Okay. Great movie. What happens, Jodie Foster is part of SETI, and there's a signal from space, and she noticed a signal from space has prime numbers, and nothing's going to produce prime numbers. And what happens is she then notices and learns with help that it has the schematics of a spaceship. So when she saw this information-rich signal, she knew it was designed. She knew it was aliens. No one's going to say, Jodie Foster, it's unscientific to appeal to design. you have to assume there's maybe self-replicating stars, and natural selection is selecting for stars that produce signals with schematics of spaceships embedded in them. Does that make any sense? No, that's crazy, but that's exactly what's happening with origin of life. What you do is in a minimally complex cell, you see information that encodes for that. And the positive evidence for design is so clear and so unambiguous that to deny it, you have to actively suppress the truth. But what do they say? Oh, not a problem. You've got these self-replicating RNAs that are being selected for that, which is crazy. So what happens is the positive evidence for design associated with minimally complex cells is really indisputable. Thank you. So. Okay, any more questions or comments? Or? Not bad. Under an hour.
Keep getting told that the best skill for a physicist is knowing how to answer good questions, and I'm clearly not there yet.
Does basic logic make sense?
Did they use the same logic, like, for the hydrothermal vents? Because I know one of the other theories is, like, the shallow warm coastal seas. Is it the same, like, issues that arise with the thermal vents that would arise with that?
Yeah. Every system, and probably the most interesting research is by people like Benner, who does, I think, these thermal vents on land. And you always get to the same problem, is that if you're lucky, if you imagine, let's say, these dehydration wet cycles, you can imagine that, you always hear this all the time, you can imagine, that's always a dangerous start, that you might get amino acids that could link together, maybe you can get RNAs to link together, or nucleotides. But the problem is always the same, that it's the end of the story, because the energy is much more likely to break things apart as to put them together. So sure, maybe you get a few amino acids that form together, but the next cycle is going to break them apart, more likely than to add on to your chain. And then the other problem is there's so many reactions that can take place. So even if you just have all nucleotides, the nucleotides can link together in different ways. So you're never going to get all the right linkages to form a functional ribozyme or a functional RNA. You're always going to get these other side chains and things like that that give you junk. So it's the same problem over and over again. You can't match the energy properly. And there's so many reactions that could take place that the life useful ones are just a very small minority. So it's always end of story very, very quickly. No problem.
On the last slide about the processes necessary for a minimal cell, you mentioned that you need a is this algorithmic cognition? So this is assuming a what does that mean?
Okay, great question. What happens is having a functional metabolism is really, really hard, because you have all these chemical reactions taking place. And what happens is you can get isolations. You can get things to get out of control. So let's imagine pyruvate builds up and keeps building up. What's going to happen to your cell? It's going to blow apart, right? So you have to keep all these things in the right concentrations. So there's tons and tons of feedback loops, like a lac operon is a classic example, where you've got feedback, where you've got special enzymes that slow or speed up reactions depending upon the concentration of these different metabolites. If you don't have those feedback loops, and that's what I mean by algorithmic cognition, you're basically sensing the environment, and then you have a mathematical analysis of that environment. You have to figure out what you want to do and then send a signal to a control system to do what you need to do, either to speed up a reaction or slow a reaction down. That's what I'm talking about. And you have to sense the environment. If it gets hotter or colder or higher pH or lower pH or less nutrients or more nutrients, you've got to control your control gates. You've got to have active transport, put more energy into maintaining a disequilibrium. You've got to shut down some pathways. You've got to open up pathways. You've got to change the tension in your cell, because you're always sensing the pressure or how much the cell membrane is being stretched, and you compensate if you need to lower the pressure or increase the pressure. So these feedback loops are essential to figure out what you need to do. So that's what I mean by algorithmic cognition. Yes?
I might be missing something, but I'm curious, and this is towards the beginning, I think, but now that I know how the presentation ends, what was the point of bringing up the higher effectiveness of systems of more free energy? I'm not quite sure how that corresponds
Say that one more time, so the importance of free energy?
Yeah.
Right, what happens is when nature is always trying to go to lower free energy.
No, I understand that part. How does that correspond to intelligent design?
Oh, right, because to keep yourself at a high free energy, you have to have lots of complex machinery that produces energy currency molecules, and you have to have information, your enzymes, that directs that energy properly to maintain the system that you want us to do. So both getting the free energy requires complex machinery that are built of proteins with information-rich molecules. The enzymes that process the energy has lots of information, and information is only the product of a mind, at least large amounts of information. And the point is, you've got to have all this machinery, because if you don't, guess what? Your cell breaks apart and turns back into simple molecules.
That's just a different level of the algorithmic cognition.
Yeah, exactly. Yes?
Couldn't you, sorry, couldn't you account for some of the probabilistic disparities, right? Saying like it's 1 in 10 to the bajillion possibility that this happened. When you consider that for us to observe life, it would only take that probability happening on any planet in the universe rather than just Earth.
That's a classic response, is you increase your probabilistic resources. So then that's exactly, like I was at a conference and that was one of the talks, is maybe we live in an infinitely large universe. And if you have enough planets, all you need is one planet where things got lucky. Sure, but when you're dealing with numbers like 10 to the power of a billion, that's a 1 with a billion zeros. The number of atoms in our universe is like 10 to the 80. So you really literally need practically a literally infinite universe for that to work. And then you get into Boltzmann brain problems and so forth. But fine, let's say you do it. Well, okay, then now you've got the first life, a prokaryote. What about a eukaryote? So what you find is the information problem is not just with the origin of life, it's really bad. But whenever you go from something to something dramatically different, you need lots of new information for new proteins and new structures in your tissues. So you're constantly getting into this problem of going from A to B. You have to have lots of information. The most greatest example is the Cambrian explosion, or about 30 million years ago. You see the first representatives of the major animal types appear out of nowhere. Boom! Over a 40 million year window, these different sub-explosions where a whole phylum comes into existence out of nowhere in the resolution of the fossil record, which is like 1 million years, which is not enough time for even the smallest amount of new information. So to get this new organism, you have to have lots of new genetic information infused in the biosphere. So you keep running into the same problems. You could argue maybe if we have a virtually infinitely large universe, it could happen once, but over and over again we see this problem of information coming from the outside into the biosphere to create new types of life. So.
But at that point you have self-replication right? At that point you have evolution,
Sure.
right? So couldn't you explain it with that those new forms of life are more effective and therefore selected for?
The problem is selection only selects for what's immediately useful. The problem is there are irreducible steps to go from A to B. One is just to get a new protein with a new fold. And the chance of a random sequence forming into a functional fold is like 1 in 10 to 100. So that step, that irreducible step, requires enormous amounts of information, which is far more than what could be produced in that amount of time. And there's a paper that was out in 2008 by Duret and Schmidt that says, how long does it take for new information to enter the biosphere? So if you have, let's say, humans and our ancestors, humans and chimpanzees were supposed to diverge about 7 million years ago. But the time it would take for, population of our ancestors, like 10,000, generation span of like 20 years, the time it would take for just 2 specific mutations to appear in an individual, 2 coordinated mutations, is like over 100 million years. Just 2 mutations. So for fruit flies, it's like 7, 8 million years for 2 coordinated mutations that are neutral to appear and spread through the population. So, and the time grows exponentially with the amount of information you need. So the problem is, before you get something like a lens, for instance, let's say you want to evolve a lens. A lens, you've got to have a developmental pathway that differentiates the tissue properly for there to be a lens. That's thousands and thousands of mutations to go from basically undifferentiated tissue, which is useless, to a functional lens. So the time required for that is vastly longer than the age of our universe. So that there just isn't enough time to produce anywhere near the amount of information you need. So that's the problem.
With that, I know a lot of the time we talk about like the plasticity of the duplication within the genome. So then you have like 3 sets, like in cyclids you have like the 3 sets of genes, and so then it allows for more possibility of mutations while maintaining the original form. What is like the argument with that?
That's the same problem. So let's say you've got a new gene. Okay, fine. So now you've got something to do something new with. So it's free to mutate. But now you're going through sequence space and you're looking for something new. That's the problem, finding something that's useful, because there's so many more sequences that are useless, that don't do anything, that don't fold, compared to a sequence that does something useful, that again, the timescales are vastly longer than what's available. I just literally did a talk on this at a conference.
It's very cool, thank you.
Yeah, so again, the math is catastrophically bad. But with the origin of life, it's most obvious because you don't have self-replication. You have to get a self-replicator. So it's very simple to go with origin of life, and there isn't quite the cultural baggage. So people are a little bit more calm if you talk about origin of life issues. So. Okay, any more questions? Going once? Okay, thank you very much.
As a closing, excuse me, as a maybe closing takeaway for everyone here, if we're engaging in a conversation with someone about like the origin of life, what is like one thing we can bring up in the conversation that is like easy to remember, something that we could like maybe from your talk?
Basically, to have life, you have to have information, because a bunch of chemicals in a soup isn't going to help you. You've got to have enzymes, you've got to have structures. And you ask them, can you create large amounts of information from scratch? And I use the soup analogy is the simplest one. If you saw a bowl of soup and it had a sentence in it, you wouldn't think, oh, I could explain that with the chemistry of the pasta or the physics of the boiling water. Large amounts of information always points to a mind. That's really simple. And then the other one I do often is this issue that everything either goes to higher entropy. Like, is it easier to keep your room neat or to keep it messy? Messy, because there's so many more ways to be messy. So things will either go towards higher entropy, higher disorder, messiness, or they go to lower energy. Does water run uphill or does water run downhill? Goes to low energy, right? But life is both low entropy and high energy. And that's not physically possible. That never happens without help. And again, it all goes back to the issue of information. Because that paper I showed you, and I'll email John some of the papers I've done, so you have all the references and so forth. And there's videos out there. There's great videos. If you want to be real lazy, just say, here's a link, watch this video. Or my interview with James Stewart. So again, it all goes back to information, because the only way to get over the problem of both the high energy and the low entropy is to have information or instructions that determine how all the molecules are to fit together. So then I'd use a soup analogy. That's pretty simple to remember. Or even more simple. If you take a cell and you put it, it dies, and you put it into a glass of water, what's the cell going to do? It's going to break apart into simple chemicals. What happens if you put in a thermal vent? It breaks apart into simple chemicals. What about a prebiotic soup? It breaks apart into simple chemicals. Do you ever see simple chemicals turn into a cell? No. Molecules never move towards life. So, and then I love the analogy again, I really, really like the analogy of the Contact. I would watch the movie Contact just once. It's a really interesting movie. But again, if you have a signal from space with the schematics of a spaceship, is anyone going to question that that was from an intelligent agent? If you've got that, I mean, energy production, information processing, selective gateways, does that look like something that might be at the bottom of your barbecue pit? Kind of that gunk in your barbecue, does that look like something that's designed? So I mean, there's a point where people have to, admit the obvious. So